Sweet intro. That was really nice. So as Megan mentioned, my name is Becky Scott. I did my PhD here, graduated a couple of years ago. I was in Cam Ainsworth's lab. And now I work as a program officer for the Ocean Foundation. So today I'm just going to do kind of a high level overview of what I do at my current job and a little bit about how my PhD here at CMS prepared me for that job. And so Megan did a great job of this for me already, but just really briefly to summarize my professional background, I did my undergrad at the University of Rhode Island. After that, I worked for a bit at the NOAA Chesapeake Bay office for NOAA Fisheries as a modeling intern and eventually a contractor doing ecosystem modeling work. I did my master's in freshwater ecology at Georgia Southern, and then here for my PhD in the MRA program. After my PhD, I did about a year in the stock assessment team next door at FWC before getting my current job at the Ocean Foundation last March. So what do we do at my team at the Ocean Foundation? As Megan mentioned, we're the International Fisheries Conservation Program, and the overall goal of our team is to advance precautionary management procedures for internationally managed fishery stocks at regional fisheries management organizations. So I know it's a little bit of a mouthful, but the way that we do that is we take a three-leveled approach to that using advocacy, science, and communications. We're a pretty small team. There's only 3 of us on this team at the Ocean Foundation, and each one of us is sort of responsible for taking the lead on one of those 3 components. But we're also strategic partners with the International Fisheries Group at Pew Charitable Trust, which have about 20 people. So working with them, we actually have quite a big and very effective team. And so I just underlined these 2 terms here because I'm going to do a little bit more of a deep dive into what I mean by both management procedures and RFMOs. First, what are RFMOs? Stands for Regional Fisheries Management Organizations. These are basically international governance bodies that are responsible for managing any fishery stocks that are occurring in areas beyond national jurisdiction or the high seas outside different nations' exclusive economic zones. So these bodies include both coastal states. So those are actually the countries whose waters are home to at least part of the RFMO managed fishery stock, as well as distant water fishing nations. So these are countries whose fleets actually travel to fish in different oceans. So for example, Japan is a distant water fishing nation in the Atlantic Ocean. And these RFMOs are established by international agreements or treaties. So you can kind of think of it, I always say it's kind of like the UN for tuna. And so there are 2 different types of RFMOs categorically. The first and sort of the major one are these, they call them tuna RFMOs or TRFMOs. There are 5 of them. I'm not going to read you out all the acronyms. There are a million acronyms, and I think everyone's scientific field, but you can see there's one in the Atlantic. There's 2 in the Pacific Ocean, one in the Western Pacific and one in the Eastern Pacific. There's the Indian Ocean Tuna Commission, as well as the Southern Ocean. And they're called tuna RFMOs because they manage tuna and tuna-like species is what they're called. So obviously all the, the key commercially important tuna species, as well as billfishes and some pelagic shark species. We also have general RFMOs, so there's about 8 of these, and these are, distributed all over the world, but these are for species that are not tuna and tuna-like species. So the species kind of vary between the RFMO, but again, these are internationally managed stocks that are being fished on the high seas. So things like Antarctic krill in the Southern Ocean, camlar, jumbo flying squid, or you might have heard them as Humboldt squid in the South Pacific, or Northeast herring in the Pacific. And these RFMOs cover over 90% of the ocean, so huge part of the world. And the TRFMOs manage over $40 billion in tuna annually. A lot going on. And just a little bit of background on why these things exist. So in 1995, the U.S. Fish Stocks Agreement was signed, and this made the precautionary approach a legal obligation in the management of any highly migratory fish stocks or stocks that are straddling international boundaries. And so this means that it requires coastal states and RFMOs to apply precautionary management for when managing these stocks, which includes mandates for reference points and harvest control rules, which I'll talk a little bit more about in a minute, as well as conservative management response when these limits of these stocks are approached or exceeded. And management procedures are a practical application of this precautionary approach. So what are management procedures? Management procedures are also known as harvest strategies, and basically what these are pre-agreed science-based frameworks for making fisheries management decisions, so things like setting catch quotas or effort limits that are designed to achieve specific management objectives. And MPs, management procedures, are typically tested using a management strategy evaluation. Again, I'll talk more about that in a bit to ensure that the MPs are performing robustly under uncertainty. So I'm going to go through kind of high level the 8 different elements of best practice MPs. The first of which are your management objectives. So management objectives you're basically using to set the goals for your vision or your goals for the fishery, where you want the stock to go. These are discussed and adopted at the very onset of sort of the MP development process. They're adopted at the management level. So managers have the final say on what the management objectives are for their fishery stock. But these are measurable goals for your fishery. And they can fall into a couple different categories. So things from maintaining the stock at a healthy level, maximizing catch stability. And so just to give you kind of an example of a management objective that you might see. So for your safety management objective, avoiding the stock size falling below 20% of the biomass that would result in maximum sustainable yield with a 90% probability over the next 30 years. So that's just one example of a management objective for a stock. Moving on to reference points. These are formally adopted benchmarks that are used to compare the current state of your stock with the desired state of the stock, which is also called a target reference point. Sorry, it's a little cut off. I'm not sure if I can do this. There we go. The desired state of your stock, also called your target reference point, or an undesirable state of your stock, also called a limit reference point. And these reference points are often specified within your management objectives. So going back to that example management objective I just gave on the last slide, we're avoiding our stock size falling below 20% BMSY. In this case, that 20% BSY would be our limit reference point, so our undesirable state. And then the last thing you sort of define in advance of even, starting the modeling for determining your NP is deciding on your performance indicators. Performance indicators are how you're going to quantify whether or not you're meeting your established management objectives. And so just some example performance indicators that could be associated with different management objectives. So for example, if your management objective is to maximize your overall catch levels, the performance indicator that you could use to quantify that and assess if you're doing that could be your total allowable catch within your first management cycle. If your management objective is to have stability in your fishery, the performance indicator that you could use to look at that could be mean variation in your total allowable catch between your managements. So your management objectives and your reference points and your performance indicators are really setting your vision for where you want to go with the stock and how you're quantifying if you're achieving it. And so now we're going to move on. Now that we've defined all that, we're going to move on to crafting potential management strategies to achieve your objectives. And so I have sort of these core components of an MP highlighted here. We're going to go through briefly one by one. The first of which is your data collection program. So this is basically your strategy for collecting all the data that you need to be able to assess the size of your stock. So this is typically already in place for a lot of stocks, especially if they've already had a stock assessment. You then have an estimation method. So this could be model-based or empirical. You're taking the data that you collected in your data collection program, and you're assessing what your stock status is relative to different reference points or indicators. And then you have a harvest control rule. I'm going to deep dive a little bit more into that on the next slide. But your harvest control rule is basically a pre-agreed rule that you're using to set your fishing opportunities based on what your estimation method determined your stock status to be in the previous step. So again, just a little bit more of a deep dive on harvest control rules because they are very key here. They're an operational part of your management procedure. So basically on this plot, the harvest controller is a sort of black hockey stick shape that you see. This is an example of a really traditional, they call it a hockey stick harvest control rule. But you can see our reference points on here. So we have our target reference point here, and you can see how they are kind of defining the shape of your harvest control rule. So if we assess our stock status to be greater than our target reference point, this be target right here, you can see we're keeping our fishing at a certain pre-specified fishing intensity. If we assess that the stock size is below our target reference point, we kind of find that, say we're at 25% of BMSY here. We follow that up on the harvest control rule, come across and use that to set our fishing intensity. If our stock status drops below our limit reference point, you can see in this example of an HCR, our fishing would close. So again, the relationship between stock status and your recommended management action are guided by your target and limit reference points typically. And this is just to show that this, again, I said, is an example of a hockey stick harvest control rule, but harvest control rules can take all different kinds of shapes. And they can be either model-based or empirical HCRs, and this kind of goes back to whatever your estimation method is. So if your estimation method is relying on a model, oftentimes they use stock assessments or simplified stock assessments. They call that a model-based HCR. If your estimation method is just using something much simpler, so say a single CPV index or abundance survey, they call that just an empirical HCR. So that would be used in situations when you don't have a lot of data for your stock, you have data poor stock, or if you don't have a reliable stock assessment available. But moving on to assessing the performance of our MPs, right? So we define our management objectives and our reference points and all that. We've crafted some potential hypothetical harvest control rules that we want to test out. And so then we're going to test out the performance of all of those MPs using a management strategy evaluation, which is a closed loop simulation testing of alternative management procedures under plausible future scenarios. And the goal of this modeling is to identify a management procedure that's robust to uncertainty and is likely to achieve our management objectives regardless of which of our future scenarios turns out to be correct. And the first step in doing this closed loop MSE modeling is constructing what are called operating models. I'll again do a little bit more of a deep dive to that on the next slide, but basically you're crafting an operating model that is reconstructing your historical fishery and then being used to simulate your fishery into the future. Operating models are considered the building blocks of MSEs. And so typically when you're doing an MSE, you make many operating models. And that's because each operating model represents a single hypothesis of the structure of your fisheries system. Like I said, they're often based on stock assessment models or other simplified assessment models. And so the reason that there's so many operating models is because to improve the likelihood of your management procedure achieving your desired management objectives, it's really important to account for all the potential uncertainties in your fishery system. So if there's any fish people in here, you probably know that some of the biggest uncertainty in stock assessment are your estimates of natural mortality, your stock recruitment relationship, basically anything you can't go out and object or specifically measure and you're estimating within your model introduces a lot of uncertainty in your model, right? And so you can craft your operating model. So say if your natural mortality is probably anywhere between maybe 0.2 and 0.3, you could create 10 different operating models, each of which has a different ramped up value of your natural mortality. And you could do the same for, say, steepness, your stock recruitment relationship. And so that ends up building you sort of a grid of operating models, each with different values quantifying different levels of your key uncertainties. And this is called your reference set of operating models. And then you can also create a robustness set of operating models, which basically represent more extreme but still plausible uncertainties. So say if you expect your growth to decrease or your recruitment to fail in the face of climate change, you can construct robustness set operating models to reflect those changes. And so what the robustness set is really good for is say all of your candidate management procedures are performing equally well and meeting your objectives under your reference set of OMs. Then you can look to, okay, well then which one of these MPs is still performing well under, say, potential climate change impacts. So we are capturing in this robustness set. So once we have our operating models constructed, we're then introducing observation error into the data, the simulated data, just to sort of reflect reality. Obviously, no surveys are perfect, so we're introducing that error there to resemble what's going on in the real world, such as bias and that should say catch, but bias to imprecise catch. And then we're applying this management procedure to our simulated fisheries data. So again, this is our stock status indicator plus our HCR that we talked about briefly. So we're using the simulated data from our observation error model to estimate the current state of the stock. And again, that can be either model-based or empirically. And then we're comparing that estimated stock status indicator with our harvest control rule to determine the management recommendation. So again, like when I showed on the HCR, we're assessing our stock status, we're finding it on the HCR, and then what the associated recommended management would be. And then you can also introduce some implementation errors. So this represents things like illegal and unreported fishing that would happen out on the water that could cause you to go over your recommended quota. And then you sort of repeat this closed loop cycle iteratively into the future. So it's typically projection periods of like 30 to 50 years. And so this closed loop simulation is for projecting the stock or fishery dynamics for every different operating model. And this allows managers to understand both short and long-term outcomes of each different candidate management procedure. And when you repeat this closed loop simulation for thousands of simulations, with you're introducing that error structure, this allows you to quantify the uncertainty in your outcome. So this is an example of a result plot that you could get from an MSE model. Each one of the thin black lines represents a single operating model. The bold black line would be the mean of the suite of operating models. The green and red dashed lines would represent your target and limit reference points. So you can kind of assess how your biomass or your stock size is staying relative to your reference points over time. And you see we've quantified the uncertainty with the confidence intervals there. And so these are some example results plots of what you could expect to get out of an MSE. So we see over 6 different candidate management procedures on this plot. We have a box plot showing how our stock status is compared to our different reference points, as well as these plots on the right. These are called Kobe time plots, but these are basically showing you, I mentioned when you do all these simulations and you have this error structure, you can calculate your uncertainty or your probabilities. And so these are showing basically the probability of your stock being within each quadrant of the Kobe matrix over time. So again, for non-fisheries people, a Kobe matrix is basically a quick way to qualitatively say whether or not your stock is overfished or undergoing overfishing. You want it to be in the green quadrant, obviously. And so these are plots that are produced to basically show you the probability of your stock staying in that healthy green zone over the projected years in your simulation. And then what's great about MSE is you can also assess trade-offs of your different MPs. So this is a trade-off plot that's showing your stock status on the y-axis as well as your mean catch on the x-axis. So you can assess how your different MPs are balancing your management objectives. So for example, in this one, we'd want to look for the MP that is still within our healthy green zone above our target reference point for our biomass, but still maximizing catch to the extent possible. And then the last step of crafting a fully specified MP is to define what's called exceptional circumstances. So these are basically rare and unforeseen events that were not accounted for in your MSE modeling or were outside the scope of the MP that you tested. These can be triggered by things like your stock trajectories deviating beyond the range that was originally predicted by your MSE model, any sort of significant environmental regime shifts. So say there's like a really extreme ENSO event or something, or maybe a deep water oil spill that really tanks your recruitment, something outside the range that was considered within your model design. These are reviewed annually for each MP that's been adopted. And so alongside a management procedure at these RFMOs, they're also adopting what's called an exceptional circumstances protocol alongside the MP, which are pre-agreed actions. So say we review exceptional circumstances annually and we decide that something happened that was outside the range that we tested for. This exceptional circumstances protocol will outline specifically what action needs to be taken, whether or not that's going back and revisiting the MP or things like that. Okay, so now that you have sort of a high-level background of what an MSE is and what HCRs are, I wanted to include a couple slides here showing how what I did at my PhD at CMS helped me get this job. And so my dissertation here was really focused on pelagic feeding dynamics in the Gulf of Mexico. And I was particularly focused on the trophic interaction between large pelagic fish like tuna and vertically migrating mesopelagic prey. So we have some myctophids here. And so that was the whole point of my dissertation, was that these large pelagic fish really love eating these vertically migrating deep water species and that they provide a really reliable source of food for them. And so for my third chapter, I used our Atlantis ecosystem model as an operating model in a management strategy evaluation. And I tested multiple different harvest control rules that adjusted the maximum fishing mortality of our predator, which was the yellowfin tuna, based on what the biomass of the mesopelagic fish was. So it was multi-species HCRs. And just some of the main conclusions from that chapter was that all of the candidate harvest control rules consistently yielded higher yellowfin tuna biomass and catch than a constant fishing strategy. So adaptive management, or changing your fishing intensity based on the biomass of the prey, consistently yielded higher biomass than if we were just to keep fishing at a constant level the whole time. These ecosystem-based harvest control rules that considered the prey biomass outperformed traditional single species-based harvest control rules. And the benefit that the ecosystem-based harvest control rules provided over the other management strategies increased as the biomass of their prey decreased. So in other words, the less prey they had in their system, the more important it was to manage with an ecosystem-based approach. So that's kind of what was on my CV anyway that made me attractive to my current job was this work with HCRs and MSEs that I did at CMS. And I actually, one of my committee members attended RFMO meetings. And so my committee member got me involved in presenting some of this work at some RFMO meetings, which was a great experience. And then one last little bit of background info. I shouldn't say background info, this is the whole talk, but high level info. It's just a lot of you might be wondering, if you're fish people, how this compares to traditional stock assessments. So for the last 50 years, this has been the traditional management approach of RFMOs, is to do a stock assessment for the stock and use those, the advice coming out of those stock assessments to set their rules and regulations. And that's typically done with negotiations between all the member countries within an RFMO. And so this can have some challenges, especially in these really complicated international multi-member fora. And so one challenge of this is true for stock assessments in general, but there's typically no consideration of how resulting management decisions might perform if you underestimated the uncertainty in your model. So again, stock assessment really focuses on trying to most accurately represent reality in a single model, right? But if you're underestimating your uncertainty, this can lead to management advice that could either over or underutilize your stock. So we always like to say we are an NGO, but I'm equally concerned if we're underutilizing a stock than if we're overutilizing it, because we also really care about fishermen livelihoods. So that's not good either. And specifically at RFMOs, this is a big limitation, is that the advice that comes out of a stock assessment that's being given to managers by the scientists is then negotiated. So it's not taken at face value. It then goes into this really lengthy political negotiation process where countries are arguing back and forth over who gets what quota. This can lead to stalled negotiations and no management whatsoever, or it can lead to short-term decision-making based on political decisions and considerations instead of the actual science. So you can actually end up fishing over what was recommended by the scientists just because of these sort of messy political negotiations. And then the focus on stock assessment is typically on setting the fishing levels for the next management cycle. There's typically not much reflection on whether or not what you did in the last management cycle actually met the management objectives that you wanted them to. And so this hasn't been the most successful management approach for all global fishery stocks. This is from an FAO report that came out in 2024, which is basically showing that the fraction of fish stocks fish at sustainable levels has decreased by about over 25% in the last 15 years. Whereas advantages of management procedures, again, it's very robust to uncertainty. So like I said, we're capturing a range of uncertainties in our operating model, and we're finding a management procedure that's still meeting our objectives over a potential range of uncertainties, right? So we don't know what the true state of reality is. That's not the point. The point is to find a management procedure that still performs well, regardless of what it actually is. And this is structured decision-making. So MPs are pre-agreed upon rules. You've already agreed to that harvest control rule. And so it helps you set really clear decisions and reduce political negotiation. So if all the countries already adopted this harvest control rule, they've already agreed that whatever our stock status is assessed as, we go up on our harvest control rule, we go across, and that's our fishing quota. It's also a much more inclusive process than traditional stock assessment. So stakeholders are involved in the whole process of the MPs. Every RFMO has what's called a science management dialogue group that's open to all interested stakeholders that meet several times throughout the MP development process, from things like setting management objectives to performance indicators and selecting the final management procedure. And like I showed you with the different categories of management objectives, MSE and MPs can integrate multiple management objectives, so things related to both biology and socioeconomics. Like I said, this is often collectively established with stakeholders, which allows them to actually buy into the management procedure. Sometimes you can get really angry fishermen with the results of stock assessments, but you can prioritize fishery stability, which is great for fishery livelihoods in MPs. And so, yeah, allows for the consideration of trade-offs between different management objectives. So overall, MPs represent a shift away from this short-term reactive decision-making and more towards a pre-planned approach. Yeah, so greater predictability for fishing industries and effective management. And I'm going to skip this really quickly because we're short on time here, but just to say that if any of you have ever seen this sort of blue logo on the seafood that you buy in a store, this is the Marine Stewardship Council logo. And essentially, fishermen really want this logo on their product because consumers actually really care about buying sustainable seafood. And in order to get that symbol now, with the new standards, that fishery to be certified by MSC actually has to have a fully implemented management procedure in place to maintain their certification. So industry actually want management procedures to be adopted as well, so they can get this certification. And just say that this is actually very widespread in RFMOs now. So these are, there's MPs currently in place for 11 of the 23 commercially important tuna stocks, as well as 2 different swordfish. And there's a lot more in development that are expected to be adopted in the next 3 years. And it's not just for tuna. So there are MPs in place for other stocks like small pelagics and demersals around the world. Okay, so I know I'm a little over on time, but I just want to wrap this up by giving you just examples of actually how we are advancing management procedures or harvest strategies in this RFMO space. So basically what specifically my group does to get the management procedures across the adoption finish line. And so I showed you this triangle before. How we do that, we take this 3-tiered approach of government advocacy, science, and communications. And so what that looks like in terms of advocacy, so we attend a lot of international RFMO meetings between our Ocean Foundation team and the Pew International Fisheries team. We are involved in almost every RFMO on the world. And so on any given week, someone's on the road at an RFMO meeting. And this involves developing and fostering international relationships with contracting parties or delegations of these member countries. So developing trust with them, getting them on board, because at the end of the day, they're the ones that are voting to adopt these processes. So it's really in our best interest to foster these relationships with the different governments. And sometimes that can even look like ghostwriting proposals for them. So more small developing nations that are not comfortable putting forth proposals in the floor of RFMOs will actually help them draft their position statements, draft their technical proposals on the floor, and move forward that way. And so we actually do a lot of behind the scenes work for governments in these spaces. And so just to give you kind of an idea of how widespread we are, this map on the bottom is everywhere that I went in my first 6 months on the job. So we're constantly on the road. For communications, we are responsible for this website, harveststrategies.org. My team runs that with funding from all of those sources on the right, including FAO. They're one of our major funders. But this website is really great. I actually used this website when I was studying for my comps, so it's kind of crazy full circle that I work for this company now. But we have fact sheets, animations on what harvest strategies are, what MSE is. We have a quarterly webinar series. If you go on our website and want to sign up, we've done topics like multi-stock MSEs, involving stakeholders and local communities in MSE development, and MPs in climate change scenarios. This is what I am most excited about. We recently launched an e-learning course with FAO on management procedures in MSE. It's 5 modules. It's about 4.5 hours of total learning time. But if you take it, you get a certified FAO e-learning badge, which in some developing countries actually can help people get promotions within their jobs. So we're really excited about that. It goes a long way for increasing capacity in different countries to actually conduct MSE modeling themselves. And it's a really cool course. So like I said, there's 5 different modules. The 4th one is actually this fun sort of simulation training adventure where you pretend that you're a fisheries manager and you go through the whole MP development process all the way through selecting your final management procedure. We put on capacity building workshops for industry managers or science or other interested stakeholders. Again, that's just to increase capacity. MSE is kind of a complex modeling topic. And so the more people we can get to fully understand the process, the more likely we are for people to feel confident voting on it and adopting it on the floor of RFMOs. So we have one coming up in a couple weeks in Egypt for a bunch of African countries that are involved in the Atlantic and Mediterranean RFMOs. In October, we put on one in the Seychelles for the Seychelles Fisheries Authority. For science, this is why I was hired. So we're trying to increase our internal capacity to be able to actually do some of the MSE modeling. We work really closely with this consulting firm based out of Vancouver called Blue Matter Science, who developed this open MSE software platform. So I've been training with them. I'm going to be involved in the modeling work for the upcoming North Pacific Swordfish MSE. And we also will fund external capacity to conduct the MSE. So we provide funding to Blue Matter scientists or other country scientists to do the MSE modeling to actually get it across the finish line. And then finally, we host a series of professional workshops. We held one in October in Rome on blue shark MSE. So we brought together blue shark and MSE experts from all over the world and talked about how we can develop and implement MPs for blue sharks at all the different RFMOs. And we have an upcoming workshop at a joint ICES-Pisces workshop in May on operationalizing ecosystem-based management of forage species using MSE that me and my coworker at Pew are convening. So that's it. Thank you for letting me go a little over time. but I will take any questions. We're going to get started again. As a reminder, some of the physical feedback forms were passed out. They were also sent to your email, all CMS email. If you choose to fill them out online, just email them back to me and I'll make sure that they get back to the person. Otherwise, you can bring them up to your physical feedback forms. You can bring up to Alexandra and I up in the front. So our first speaker today is Liliana Gifrida. She's a master's student with Dr. Brisbane, and she's going to be talking to us today about pyridinium.
Hi everyone, thank you so much, Emma, for the introduction. My name is Liliana and I'm in the MECO lab led by Dr. Maggie Brisbane. And today I'm going to be talking to you about a project that we've been working on the past several months, which is untangling the impacts of bacterial interactions in changing environmental conditions on the growth and toxin production of pyridinium bahamense. So for some background, who is pyridinium bahamense? It is a Eurohalien subtropical tropical dinoflagellate that is found in estuary waters all over the world. It is also a harmful algal bloom or HAB-forming dinoflagellate. There are two known variants of pyridinium bahamense. There's var compressum and var bahamense. Compressum is typically found over on the Pacific, and then var bahamense over on the Atlantic side. So that's what we're going to be focusing on today. It was originally thought that var compressum was the only variant that could produce toxins, but now var bahamense has been found to be a saxitoxin producer in Florida estuaries and cause paralytic shellfish poisoning. In terms of their physiological limitations, they can withstand temperatures between 20 and 32 degrees Celsius, but their ideal bloom temperature is around 25 to 30. They can also tolerate a wide range of salinities between 10 and 45. Their vegetative or their photosynthetic swimming state is typically found in low concentrations through the spring, with the blooms happening in the summer. Pyridinium is also assisting dinoflagellate, meaning that they can enter into a cyst phase, which is in this bottom left image here. Well, they'll go down into the sediment when conditions become unfavorable. And areas with high residence time are more optimal for their growth. So it's not surprising that pyridinium likes Tampa Bay because we have a lot of nice bridges that restrict the flushing rate in this area. It is really important to consider the bacterial interactions with dinoflagellates because they can either be friends or foes, and that they can be symbiotic or competitive for the same resources. We know that HAB dinoflagellates need a lot of B vitamins, particularly B12. So pyridinium will sometimes rely on symbiotic bacteria in the phycosphere for B12 supplementation when the concentration in the water is limiting. But as I said, it's not always a nice relationship. Sometimes bacteria can be opportunistic or even algicidal towards pyridinium. So with all of this in mind, our objectives of this study was to untangle the impacts that the changing environmental conditions might be having on pyridinium growth in toxin production, but we also want to see what role the bacteria are playing in that, if any. So to start, an isolate of pyridinium bahamense, var bahamense, which I'm just going to refer to as pyridinium for the rest of this talk, was isolated as a cyst from Old Tampa Bay by the FWC. And from this subculture, we had an attempt to create an azenate culture, and from this attempt, we saw that no bacteria grew in a high nutrient test broth, and there was little microbial diversity. So from this subculture, we created two isolates. The first isolate, which is on the left, retained a low diversity microbiome, and so there's not much different types of bacteria here. And then to the second isolate, we added surface seawater from Old Tampa Bay to introduce a more diverse microbiome for the pyridinium to recruit, and that's the second isolate that we see on the right-hand side. We wanted to do two different isolates with different microbiomes because we wanted to really see how the diverse bacteria might impact their growth and toxin production. So to show you guys our experimental design, it can be a little bit complicated, but to start, if you recall the two different isolates that we had, the two different microbiomes, we have the low diversity and the high diversity that had 36 flasks each. And then from 12 of each of those 36, 20 were at a salinity, or 12 were at a salinity of 20, which is our low salinity. 12 were at a salinity of 25 for our medium. And then the final 12 were at a higher salinity of 30. Then we separated these again by nutrient additions. So 6 of each of those 12 were amended with nitrate and ammonium, and the other 6 were amended with only nitrate. And then the final separation was by temperature. So half of all of those triplicates were put into a warm incubator set to 32 degrees, and the other a cooler incubator set to 24. So after all of these separations and replicating, we result in 72 culture flasks that represent every possible combination of the parameters that we have here. So when we get to the analysis portion, we can see what might be causing those changes. And we repeated this process for 3 trials, but the nutrient variable wasn't added until trial 2. Every 48 hours, we ran 2 96 well plates through our attuned CyPix flow cytometer. One of those plates was for pyridinium cell counts, and the other was for bacterial cell counts. We also obtained images to look at morphology data from our attune, and then we measured chlorophyll A fluorescence as a proxy for pyridinium biomass every 48 hours as well. And like I mentioned before, we had 3 trials overall, and each trial was a serial transfer from the previous. Trial 1 was a preliminary trial. It ran for a shorter amount of time and did not have that nutrient variable. We added the nutrient variable in trial 2, and then we did a transfer to trial 3. So when trial 2 concluded, we filtered down what was left in those flasks and filtered them for RNA extractions, Illumina short read sequencing, and ASV analysis, so we could see if all of those treatments were affecting the microbial community composition. And after trial 3 concluded, we also filtered down what was left in those flasks for saxitoxin analysis, which was done using HPLC by our collaborators at the FWC. So these are the results from our bacterial counts of trial 2 and trial 3. I'm going to be using this style of plot a couple times in this talk. So just to point out some key features, we have our nutrients on the top of both, and then they're separated by salinity, and then the temperature. The cold ones are here. This is the 24-degree incubator, and then the warm, which is the 32. And a key takeaway from this figure is that both microbiomes reached higher bacterial cell concentrations in the warmer incubator, showing that they really thrived at this temperature. And consistently throughout, regardless of salinity or temperature, the OTB, or the high diversity microbiome, consistently reached higher values as well, but they both thrived better in 32-degree incubator. And as I mentioned before, we filtered down the flasks from trial 2 to look at the microbiome, and this was our sequencing data. So these 2 on the left was our low diversity microbiome. This is 24 degrees, this is 32. And then this is our high diversity OTB microbiome, 24 and 32. And at first glance, we can see that the 2 on the right are more diverse, but if you look a little bit closer, there are 3 groups, which can be a little bit difficult to see, but there's these dark blue here, these like light pink here, and then these like orange ones down here. And these are groups that we only saw pop up in larger concentrations in the 32-degree incubator, but they're barely detectable in the colder ones. So this is leading us to believe that temperature might have something to do with the bacteria that are more successful. And the temperature differential for culture microbiomes can be a little bit easier to see in an ordination space like this. So we have our low diversity on the left, high diversity on the right. And regardless of the salinity, we can see that we have a clear temperature differential splitting up these 2 plots. So these are our results for the pyridinium biomass that was obtained from our chlorophyll A fluorescence measurements. And we can see that in higher temperatures, pyridinium in the low diversity microbiome did better than the ones that were grown in the high diversity microbiome. And that's represented by the lighter colors, and the higher diversity microbiome is the darker colors. And we ran a multi-way ANOVA to assess the growth rates between different parameters. And we found that statistically, microbiome by temperature was significant throughout all of the trials. And this is just the statistics from trial 2, but I have all the other trials if you would like to see them after. And something else that we noticed was we had the 2 different nutrient amendments, but we didn't really see any clear patterns distinguishing the 2. So this prompted us to do an ammonium trial because we weren't sure that pyridinium could even take up ammonium directly, and maybe that's why we weren't seeing a big difference when we gave them ammonium as well. But through this trial, we saw that pyridinium can take up ammonium when it's the only source that it's given. So after confirming that they can take up ammonium, we wanted to compare the maximum growth rates between when we're giving them nitrate versus when we're giving them ammonium to see if maybe they have a preference, which is what you can see in the box plot on the right. So in order to do this comparison, we took the nitrate-only replicates from the previous trial 2 and 3 that I showed you before, from the same salinity and temperature to compare them to our ammonium trial. And we found that the pyridinium grown in a low diversity microbiome, which is represented by the yellow, performed better and had faster growth rates when given ammonium. But the pyridinium that was grown in a high diversity microbiome performed better and had faster growth rates when we gave them nitrate. So that was just an interesting result that we didn't anticipate seeing to start. The most recent development from the study is the saxitoxin portion that was done by our collaborators at FWC. So upon first glance, it might be difficult to see any trends, but we didn't really see any environmental conditions that had a large impact on toxin production. But when we look at the two different colors here, the high diversity is the darker color, we know that this data suggests that the pyridinium that's grown in a high diversity microbiome is producing more toxin per cell. And this indicates that there might be some regulatory role that bacteria are playing on pyridinium's toxin production. So some of our conclusions thus far is that pyridinium cultures in a high diversity microbiome perform better at low temperatures and low salinities. And this goes hand in hand with the second point that at higher temperatures, the higher diversity microbiomes might be competing with pyridinium for resources. And that's what we saw when we saw that the higher diversity bacteria were really thriving in those warmer temperatures, that maybe because they're thriving, they're suppressing the growth of the pyridinium. And lastly, again, we found that pyridinium grown in a high diversity microbiome was producing more toxin per cell, and there might be some kind of regulatory role that the bacteria are playing there. Some future directions that we have for the study is I mentioned before that we took some images from our flow cytometer, and in the interest of time, I didn't include them in much detail, but these are some of the images that we took from the flow cytometer, and we weren't seeing any clear patterns with certain morphologies in certain conditions. So we're not really sure what's causing them to look in these various ways, but that's something that we're interested in pursuing. And if you recall the box plot with ammonium versus nitrate, we are curious about how the microbiome composition influences how effectively and efficiently ammonium can be taken up by pyridinium. And that is all I have. Thank you for listening, and I can take any questions.
Thank you everyone for coming. This is part of my research. This is one of my chapters that is surface temperature in coastal ocean circulation, influencing tropical cycle and rapid intensification, as reviewed by 27 years of work observations. So as we know, the coastal oceans are the last few miles before a tropical cyclone makes landfall. They always experience extreme winds, storm surges, and compound flooding. So it is very important to determine whether a tropical cyclone is intensifying as it leaves less time to prepare and evacuate. And as we all know, almost every year we get the experience of the passage of a tropical cyclone here in our backyard. So here I'm showing the recent major hurricanes that have made landfall in Florida in the last 7 years. So recently, Leo and colleagues, they describe Hurricane Ian's rapid intensification in relation to anomalously warm subsurface waters on the West Florida shelf. So here I'm showing part of the results. So these are subsurface temperature observations from multiple war areas that we have. So here, basically, we have to pay attention to the red time series. This is the temperature observations at 10, 20, 27 meters of depth, and the black thick line represents the mean climatology. So this is telling us that while Ian was approaching to Florida, the water was warmer by even 3 degrees Celsius. So this is anomalously warm, and this leads the rapid intensification. Also, besides using these more observations, they rely with the absence of a loop current interaction that I'm going to explain right now. So essentially, Liu and colleagues in 2016, they described a loop current interaction with the West Florida escarpment here near Dry Tortugas called the pressure point. So if this interaction persists, it can set the entire shelf in motion, inducing a boiling, and we have now colder waters. And our lab has a great opportunity to study the West Florida shelf with multiple observing systems, and one of them is COMS. COMS consists of a surface and subsurface mode array, providing real-time observations of temperature, salinity, and ocean currents. So this is ideal if we want to capture any thermal changes before, during, and after a tropical cyclone. So we are following this hypothesis that with the absence of this interaction, we have warmer waters at subsurface, and this leads to the rapid intensification on tropical cyclones. We have two cases. One, when tropical cyclones rapidly intensify on the shelf, and when they don't experience any intensification on the shelf. So I'm going to explain first. The first case is a big figure and has a lot of information, so I'll do my best to explain everything. So first, each column represents 1 year. So we have 2004, 2022, 2023, and 2024. And the first row represents the surface observations, while the second, third, and fourth row represents the subsurface observations. The last row represents the number of days on which this loop current interaction occurred in each month and each year. So the gray time series represents observations from previous year, just to have as a reference, with the red time series represents the temperature observations for each year. The black thick line represents the mean climatology. This is the, I don't know if you guys from the back can see, but this is the 26 degrees for the Atlantic hurricane season, and this vertical dashed line represents the precise moment when each tropical cyclone begins to interact on the shelf. So essentially, if we take a look to the surface observations, the temperature is warmer just by 1 or 1 degrees. But if we compare this with the subsurface observations, here the water is warmer by even 4 degrees Celsius. So this is telling us that all these tropical cyclones rapidly intensify when the subsurface water was warmer. And this coincides with the absence of this ocean forcing. And I'm showing the opposite case. Now, again, each column represents 1 year. For this case, we have 2016, 2018, and 2020. So if we take a look to the surface observations, we see a pretty clear seasonal cycle. We have colder waters on winter, and as we approach summer, we have higher temperatures. But we compare this with the subsurface observations, we don't see a clear seasonal cycle. Here in general, the temperatures are below the mean climatology, between the 20-24 degrees Celsius, and that is why we believe C13, this is C13 at 30 meters of depth, provides a good example of warm water being flushed over time. So you see the water warms whenever it reaches a maximum because it is continuously replaced by colder water. And this coincides with the presence of this loop current interaction. Also, we want to say that C13 is a good example when a sensor is not working anymore and we don't have more data. Sometimes the passage of a tropical cyclone close to the Moore array, or even the sensor failure can be responsible for this. But that is why, since last summer, our lab began to show real-time observations of the temperature in our website. So anyone can go to the website and see the temperature at 1, 20, and 30 meters of depth. You just have to click the buoy and just check it out. And this is also helping us. We don't have to wait to retrieve the data to see that something is wrong. And our team recently deployed 2 more buoys close to the panhandle. We have C24 and C23. So we are officially ready to monitor this hurricane season. Hopefully we are lucky again and we don't have to experience another strong hurricane. And just to wrap things up, the intention of this work is to highlight how important the subsurface temperature and the ocean circulation on continental shelf is. Here we see that in the absence of this interaction, we have warmer waters in subsurface, even warmer than surface. So this leads to the rapid intensification of tropical cyclones. And when there is the presence of this offshore forcing, the water is being flushed over them and is replaced by colder waters. So this is everything that I have right now. Thank you.
Howdy, again, my name is Jill Thor, and I am going to be talking to you about modeling groundfish, bioclimatic velocities and envelopes in the Gulf. So before we jump into the nitty gritty, I really wanted to talk about the question. And so what we're looking at in this study is understanding what covariates and at what space and time scales are essential for quantifying the bioclimatic envelopes of demersal species. And demersal species are things that live on or near the bottom, and in particular fish. So bioclimatic envelopes and understanding this is essentially quantifying the realized niche of a species, which is the n-dimensional hyperspace in which a species has a maximum abundance along. And so in the literature, you'll hear people say that they are using species distribution models or they're using habitat suitability models. And these are the same as bioclimatic envelopes, and all three can be used interchangeably. You'll hear me use them interchangeably, and it's all the fundamental idea of quantifying this realized niche. So there's lots of ways to go about doing this. A lot of people use a regression-based framework, and there's one major pitfall a lot of studies fall short to is that regression-based frameworks are inherently correlative. So if you can correlate the number of beanie babies produced in a year and the number of hurricanes that hit the Gulf and find a significant correlation, that doesn't mean it's meaningful, but it might be significant. So as biologists, ecologists, and scientists, we have to do a good job of figuring out what is a meaningful covariate when we're going to model a fish and figure out what's going to drive their abundance. So again, what's available isn't always best. And we have to remember that too when it comes to scale. Like what space and time scales are important. So who cares? And why does it matter? Well, these models are really the foundation for how we assess fish movements, especially with respect to climate change. So these are the models that allow us to figure out if species are shifting in response to warming temperatures or different climates. And most studies of this nature have been done in linear latitudinal coastlines like the Atlantic, where there is a single poleward gradient and the fish are just following that gradient. What happens when you enter a semi-enclosed sea in which fish can't follow a single gradient? Let me introduce you to the Gulf. We have a poleward landmass that restricts range shifts. And a lot of previous work that's been done here are big parts of large meta-analyses in which large amounts of data are ingested. And when you have that, sometimes you miss a little bit of the nuance in what's happening in the system. And so previous studies that have looked at species shifts in response to climate change have been these meta-analyses that have looked at all basins in the US. And they've said, you know, fish are all moving and responding to climate change except the Gulf. In the Gulf, things aren't really happening. And if species are moving, they're moving in a longitudinal way, east-west, and that's about it. But when you start digging into that and start questioning that and saying, is that meaningful if it's the same species that's moving in the Atlantic but not in the Gulf? You start to see that there's data limitations. I mean, when you're ingesting huge amounts of data, you sometimes miss little like nuances. And so that really comes into this be really cool to do a PhD project on teasing out all that nuance and figuring out what's meaningful. So how do you do that? Well, we look first at the physics of the Gulf, right? We look at what's driving, what's changing in our bottom temperatures. So the first part of this project was to look at large, fine scale and a large assessment of bottom temperature change. So that's what you see here. This was my first chapter. Just to give you an idea, this is the Gulf at 1/25th degree spatial resolution between 1996 to 2012. And it was recently published in Fisheries Oceanography if you want to check it out. And we're not going to talk about that today, but the two big things we want to hit on here that leads into what we're going to talk about today is first, what Siria said too, sea surface temperature does not equal bottom water temperature. And so most studies have used sea surface temperature to figure out what a fish is doing. And that works in the Atlantic, that works in a linear longitudinal coastline. However, in the Gulf, it doesn't work. That's because we have this heterogeneous signals that are related to subbasin morphology, which means that we have isotherms that are not shifting polewards, but they're shifting in every possible direction. And so if our first principles are fish follow isotherms, it makes it really complicated when our isotherms are moving in every different direction. So this is what we found out. And so the next part of this was to figure out, let's bring in the fish and figure out how they might be responding. So we do that using the CMAP groundfish trawl surveys, which are the longest fisheries independent data series collected in the Gulf. And we're looking at the latex region, which is Louisiana, Texas. And this data series is collected in this region from 1986 to 2024. So here are all of our CMAP surveys, where yellow is the point where the survey was conducted. Reds are overlaid where we had warming signals, blues are cooling signals. And we had lots of stations there. There's a long, tedious process to clean these data. And if you want to talk about that, I would love to afterwards. We just don't have time right now. But lots of focus was given to try to reduce some of the ambiguity and the signal that might induce variation that wasn't actually meaningful for this project. So with that, we then, just to give you an idea, we had over 800 species on these surveys. And the goal is to build individual bioclimatic envelopes for each species. The hard part is when you're doing these models, you need to have enough data, and these data are inherently zero inflated. So we focused on just the most abundant species. And through a long process, I aged every record that we have. And so we could split our species into juvenile or young of year and adult species, because we know that they're going to have ontogenetic shifts. So in total, we have 25 unique species split into 35 unique life stage combinations. And to remind you of our question, it's what covariates, not what space and time scales are essential for quantifying bioclimatic envelopes. So remembering that this is inherently correlative, we have to go through and do a deep literature dive on what variables actually matter. And so these are just a few that were found and were used. We know temperature is going to drive fish distribution, salinity, bottom oxygen, vertical velocity, which is a proxy of welling. And so all of these variables are found at multiple space-time scales, or function over multiple space-time scales. And we have high resolution data in which we can pull that information in and build models off of this. So some of these are based on what data is actually available. For example, oxygen is only available at the day and site currently, but bottom temperature can be available at daily resolution, all in site, all the way to 125th degree resolution, which is about 4 kilometers, up to the entire latex wide. So this is a lot here. What you really need to know is that we took our data, we split it into training and testing data sets, and then we built models for each single covariate, because there's over 108 covariates per species, just to filter down from those 108, what are actually meaningful covariates? Okay, they're based on literature important, but what's important here from what we can tell based on the deviance that they can explain in a model. So this was our covariate pre-selection process. If you have questions about this, I'd love to talk more. And this was done for each species and each covariate. And today we're just going to talk about Layne snapper and their responses with bottom temperature and bottom salinity. Layne snapper, to give you an idea, they are a recreationally and commercially important species. And what you're looking at right here is a density plot where yellows represent all habitable depths. So anywhere that red Layne snapper may be found, where this is our temperature density plot, this is our salinity density plot. Yellows are anywhere that a Layne snapper could be found, and reds are where we actually see Layne snapper present. What you can see is there's real peakedness in bottom temperature as well as salinity, which indicates that the species is likely a specialist, right? They have some type of habitat preference. You can't just put them in a boiling pot of water and have them survive. They're going to notice. So we go through that model pre-selection process. And what you're looking at right here is of all those temperature covariates at all the space-time scales, these are the 4 that explain over 10% of the deviance. And to give you an idea, again, this is bottom temperature where you have day and week spatial or time resolution. And this is 125th degree space resolution, which is about 4 kilometers. and 1/4 spatial resolution, which is about 11 kilometers. And so it's really interesting. What you notice is that regardless of the space and time resolution, there's a peak at about 27 degrees for each space time, meaning that basically it's a redundant response. So we're seeing the same response for Layne snapper at all space-time scales. Now, when we consider salinity, only 2 had a greater than 10% deviance explained, about 1/4 degree spatial resolution and month and seasonal resolution. So daily was not important. But what we find here is the same thing. There's still peaks at the same times for both. So to take things home, first, the Gulf is really complex. It has that unique geomorphology that induces lots of regional variability in bottom temperatures that have to be considered when we're building these models and quantifying our niches. Second, unfortunately, we are at a place where we have to think about what surveys to cut. And as we're doing that, let's use science to figure out what should be cut. And if we can, it's expensive to build these regional downscaled models. So for fisheries aspects, we can help identify what space and time scales we need a downscale for fisheries purposes. Here with Layne snapper in particular, we found that mid-grain space and time scales were the most important for describing realized niches. I should note that this process was done for all 35 species. And then we also built full models to quantify those realized niches. And why is it important? It's because we've built these bioclimatic envelopes. So now next for chapter 3, we can predict where the species are moving and if it actually is a climate response. So with that, I'd like to thank you for your time listening to my fish map, and I'd like to take any questions.
Hi everyone, my name is AJ. I'm a first year's master's student here at the college in the labs of Dr. Munoz and Dr. Breitbart. And today I'm going to be talking about some of the work that I did as a research technician prior to joining the college as a student. So this is characterization of contaminants of emerging concern, or CECs, from wastewater effluent, and remediation by iron and aluminum electrocoagulation. So I'm going to start my presentation with a bit of background about our study site, the wastewater treatment plant. So wastewater treatment facilities take in water from our showers, toilets, and sinks. They treat it in a multi-step process and turn it back into reclaimed water for us to water our lawns and gardens with. Any extra water that's processed and treated but not needed for the reclaim system can then be discharged as surface water into our lakes, streams, or in this case, Tampa Bay. So current advanced wastewater treatment facilities can treat human waste, bacteria, and nutrients, but most lack the costly, energy-intensive techniques needed to remove chemical contaminants, which brings us to the question, how do we remove them? So there's a lot of wastewater treatment techniques out there, but today I'll be talking about electrocoagulation. And here you can see a figure showing what an electrocoagulator looks like. As the name suggests, an electrocoagulator utilizes electricity to coagulate particulates and dissolved chemicals into a flocculant and sludge phase. The process of applying this electricity to the electrocoagulator strips metal ions from the anode, which are then positively charged, and these ions bind to any negative or partially negative ions, chemicals, or particulates within the unit. This forms our sludge and flocculant, which can then be filtered off. As far as our electrode materials go, iron and aluminum are the most common metals used because they're cheap, non-toxic, and easily available. And as I said previously, the flocculant and sludge made in this process can then be filtered out and disposed of. So what does an actual electrocoagulator look like? Well, here's 2 pictures of 2 very different electrocoagulators. On the left is a picture of me with the electrocoagulator used in this project. This electrocoagulator can treat roughly 1 liter of water per minute. And on the right is what you might find in a wastewater treatment facility. This one can treat 600 gallons per minute. So now that I've given some background, let's dive into my experimental design for the project. So firstly, I wanted to keep this project as true to real life as possible. So this meant that unlike previous studies, I didn't create my own wastewater in the lab or spike my samples prior to treatment. I wanted to see what effects the electrocoagulator would have on water straight from the source, as if this were part of the wastewater treatment plant already. So to do this, I sampled the effluent at the treatment plant, which is the exact same water that's discharged as surface water into Tampa Bay. And it looks kind of like that. As far as emerging chemical contaminants go, I looked at 82 individual compounds across 3 main compound groups, these being polychlorinated biphenyls, PCBs, organochlorine pesticides, or OCPs, and phosphorus flame retardants, or PFRs. And I'd like to point out that some of these are PCBs and some of our OCPs are actually contaminants of known concern. We already know they're bad, but they're included in this study. I also wanted to compare the 2 most common electrode types, iron and aluminum, to see if there were any differences in treatment between the 2. So for each metal electrode type, the same method was followed. 8 samples were taken from the wastewater effluent tap at the wastewater treatment facility. Half of these were filtered and set aside, while half were treated, left to settle, then filtered. From there, I performed liquid-liquid extractions to separate out our emerging chemical contaminants from the water. I concentrated them down in preparation for gas chromatography mass spec, or GCMS. And then I ran these samples through the GCMS, which separates out these compounds and quantifies them. I did this using a method developed by Dr. Isabel Romero for simultaneous detection of CECs. So, what did I find? So I'm going to start off by talking about the results of the iron electrode experiment. And as a reminder, the goal of this experiment was to see the removal efficiency of the contaminants using the iron electrode. So I'm going to start with PCBs. Of our PCBs, again, we look at 32 compounds and we found 31 of them. On the left here is the concentration that these PCBs were found in per liter of water. So this is a nanogram per liter concentration, or parts per trillion. In red is the raw effluent. This is the effluent water that's coming out of the wastewater treatment plant. And in blue is the treated. So this is what went through the electrocoagulator. And as you can see, generally the red bars are a lot higher than our blue bars. And so our treatment process appears to remove some of the PCBs from the water, but not all of them. We can also look at this as total PCBs, when this is kind of reported how the EPA would do it. So the EPA has no guidelines for chemical contaminants in wastewater effluent, but they do look at ambient water. They do have ambient water quality guidelines that they use for lakes, rivers, and the ocean. So when comparing what we see coming out of the effluent tap to these chronic criteria in this case, and this bottom one's the fresh water and the top is the salt water, we can see that in one instance, we actually had a replicate that exceeded the criteria for freshwater and met the saltwater criteria. And again, these bars are the effluent water, so it's not the actual surface water that I sampled, but I like to put it in there for context of how high these contaminants are found at. To move on to OCPs, we found 31 of the 36 compounds. Again, concentration is in nanograms per liter. And I'd really like to point out DDT. Yeah, we found DDT coming out of the wastewater treatment plant. This was banned in 1972, over 50 years ago. And we also see a more than 50% reduction, in this case, statistically significant. We also look at other banned OCPs, Aldrin, BHC, chlordane, endosulfan, and engine. Some of these were also banned back in the 70s and 80s. Some of them were banned as early as recent as 2014. In all cases, though, we do see nice removals. In this case, we removed engine below our limit of detection. We look at permethrin, which is an in-use pesticide. You might have heard about it. You actually use it as mosquito control. So this is something that we use every day, especially around here, right? We look at both cis and trans isomers, and we find good reductions in both of these. And then lastly is atrazine. Atrazine is a currently in-use herbicide. It's pre-emergent. We found this an order of magnitude higher, which might make sense, right? It's in use. It's currently the second most commonly used OCP, just after glyphosate, which you might have heard of as Roundup. We use 70 million pounds of atrazine in the US per year. To move on to our phosphorus flame retardants, or PFRs. These I had a lot fewer compounds, so I could use individual graphs nicely, but I'd also like to note the concentrations that we see these at. Right, we're at 1,000 nanograms per liter here, and sub-nanogram concentrations over here. In general, we do see nice decreases in some of these compounds, but interestingly enough, we also had PFRs that increased after treatment, right? So we believe that this is because PFRs are actually used as common plasticizers, right? You don't want your plastic to easily combust or burn. And so you add PFRs to your plastic. And so if you recall, the electrocoagulator that I used is actually made out of plastic and uses plastic tubing. So we kind of believe that this might be stripping these compounds, in this case TCEP and TPP, out of our electrocoagulator and putting it in the water. So next I'm going to talk about the aluminum electrode experiment. This was done in September of 2024. Now the iron electrode experiment was done in July of 2024. So this water that you'll be looking at now is different than the water that we sampled back in July. And this is the case even on a day-to-day basis at a wastewater treatment facility. So again, I'll start with the PCBs. We found 19 of the 32, so we found fewer PCBs this time around. We also found them at very low concentrations. We're not even hitting a nanogram per liter here. In most cases, we only found PCBs in one replicate, maybe two. In all but one case, we fully removed them below a limit of detection. For OCPs, we found them at similar concentrations. But for the most part, they were at 1 to 2 nanograms per liter. We do still see some nice decreases using the aluminum electrode. In some cases, we do see increases. But overall, it does seem like the electrode does its job. And again, for atrazine, we found this at roughly 5 times lower concentration than we did in the previous study. And we find about a 20% reduction. And if you recall, the iron electrode had about a 50% reduction. For PFRs, we also find them again at a wide range of concentrations. We found fewer PFRs. We found a decrease with the aluminum versus the iron. So in this case, we had 5 PFRs that decreased and much more that increased. In some cases, dramatically too, right? This is about 250 nanograms per liter. And then in one replicate, we had almost 3,000 after treatment. And even in some cases, we didn't find any of this particular TPCP PFR in our effluent water, but we then do find it after we treat it with the electrocoagulator. So some concluding thoughts here. The electrocoagulator treatment does show removal of our contaminants to varying degrees. There are differences in removal between the two electrodes, right? Especially with atrazine are currently in use. The iron electrode removed 50%, and the aluminum electrode removed 20%. For me personally, doing this, we found CECs and wastewater effluent in the numerous, and in my thought concerning, right? These are old banned chemicals in some cases. They're more than 50 years old that we're either ingesting and putting into the wastewater treatment system, or maybe washing off of ourselves and they're entering the wastewater treatment system. But then remember, we use this water as reclaimed water. So all these pesticides and herbicides, we're then turning around and spraying on our lawns and gardens. So for some future directions, I'm going to be hopefully using this data set in multivariate analyses and looking at differences between compound groups. I also have the flocculant data, right? So we make those solids in the treatment process. I have that worked up, and it'll be really interesting to see if that relates to the decreases we see. Are we actually removing these compounds, or are we just putting them in the solid phase? And then I'll be continuing my work with emerging chemical contaminants as my master's project, but moving from wastewater to oysters. So thank you. Do we have any questions?
Hello, everyone. I'm sure most of you know me by now, but if you don't, my name is Alexis, and I'm in this lab. If you've heard about the shelf project before, you probably know that we're collecting eggs from the shelf and DNA barcoding those. But I actually want to share some data that I've recently been analyzing from the first year of sampling, summer 2024 through spring of 2025. So I think the first question I should probably get into is why even study fish spawning in the first place, right? Why is it important? So for many fish species on the West Florida shelf, spawning timelines are kind of approximated by appearance, or even behavior or the surrounding environmental conditions of adult stocks. More recently, larvae has been used to kind of approximate some of these timelines, but larvae can often travel hundreds of kilometers from where they respond, making them a less reliable proxy. The West Florida shelf is an area that's rich in its biodiversity, especially pertaining to commercial and recreational fish species, which makes it an important study area for fisheries conservation. And fish eggs are a really good proxy to study spawning locations and patterns since they're solid evidence that spawning is occurring. And unfortunately, our fish eggs don't actually look like this under a dissecting microscope. They look something more like this. Now, if anyone here can tell me what species of fish actually spawn these eggs, you would save me a lot of time. So the spawning habitat and early life linkages to Fisheries project or shelf project. Phase 3 is underway and will continue the long-term egg monitoring aspect that began in shelf phase 2. And this began through a collaboration with Glenn Zappi and NOAA CMAP program in 2019. And a paper was recently published by my former mentor, Keith Keel, discussing these results. He identified eggs from several economically important species at relatively high frequencies, similar to what we're going to be looking at today. So CMAP collects fish eggs during annual plankton survey cruises in August and September, and we're still barcoding those as we receive them. But here's a table that shows spawning timelines for many economically important species. The blue box represents annual CMAP collections that are taken in August and September. And here I'm highlighting the species that are known to spawn outside of that blue box timeline. This means that we're likely missing the opportune time to capture these spawning events. And the green boxes here represent when our sampling takes place in addition to that CMAP sampling. Our odds at capturing these previously undetected species has increased with that broader temporal coverage. So the goal of this project is to increase the annual egg collections to seasonal collections. We'll have our own fish egg data from summer and winter of 2024, all seasons in 2025, and spring of 2026. It's important to note here that the fall of 2024 data will come from CMAP, which were partially sampled due to the 2024 hurricanes. So as far as our study site, we're looking at the West Florida Shelf. The red dots indicate 70 fixed stations on the shelf, which remain consistent across all sampling events to allow direct comparison among seasons and years. And here's a snapshot from our June sampling event. This shows the track that we took, and each dot represents a 30-minute transect. We collect fish eggs continuously throughout the entire cruise in case there's an area or a station we'd like to deep dive into later. So the winter 2024 data set combines a November hurricane response cruise with our regularly scheduled December cruise. Because the December cruise was only partially completed due to rough sea conditions, combining these data sets gives us a more complete picture of winter spawning on the West Florida Shelf. And this map shows the final winter 2024 sampling coverage. Red dots indicate stations processed from November 2024, and green dots indicate stations processed from December 2024. And here's all the maps showing the stations where samples are processed across all 4 seasons. Also a reminder that the fall 2024 data were collected by CMAP and were selected for processing using a spatially balanced GRTS or GRITS approach, which selects sampling sites randomly but evenly across the study area. So here's a diagram of the machine that we use to collect fish eggs. It's called the Continuous Underway Fish Egg Sampler, or COOPS. It has an intake pump that's fixed on the hull of the ship about 3 meters depth, which pulls in the water that runs through the hose and into this machine, which we call the daiquiri machine. So here's an up-close video of the COOPS daiquiri machine and a photo taken during Ocean Sciences meeting in New Orleans of a real daiquiri machine on the right. Clearly, we had too much fun at this conference. But the COOPS has a mechanical arm which keeps excess water moving out of the machine. Has a mesh net which retains any fish egg-sized particles. So any zooplankton like copepods also get trapped in that net. The sample moves through the clear tubing you see at the bottom, and all the particles are collected on a mesh filter at the very end where you see that water flowing through. That filter is taken off and rinsed into glass vials. And here's a photo of an anomaly sample from this past December that is completely filled with fish eggs. So here's a diagram. You probably, if you've ever come to my talk before, you've seen this diagram before, but it explains DNA barcoding in 4 easy steps. So first, you randomly select a maximum of 96 eggs for DNA extraction, and that's just due to compatibility with standard PCR plates. We extract the DNA from each individual fish egg. We amplify the COI gene through PCR using a universal fish primer cocktail. We ship any positive products to TACGen where they are Sanger sequenced. Primers are trimmed using Genius, and products are identified using barcode of life data systems to at least a 97% identification match. So first, I want to throw some quick numbers at you from summer 2024 through spring of 2025. So about 3,300 eggs in total were collected across all 4 seasons. This includes eggs that were not picked, so over that 96 egg per sample limit. About 2,400 eggs were sent for sequencing. Of those, 2,200 eggs or so were successfully identified. 641 eggs were identified to the species level. All others were identified to the genus level, with 132 unique taxa. So here's a breakdown of the data shown as a stacked bar chart organized by family. So not only does summer have the highest egg abundance, it also shows greater family level diversity and dominance. Multiple families are contributing large proportions. This suggests peak spawning activity and broader taxonomic participation in spawning. The top 3 families for summer were Carangidae, which are your jacks, pompanos, and relevant to us, SCAD, if you know, you know. Scombridae, which are your mackerels, bonitos, and tunas, and Legenidae, which are your snappers, of course. So fall shows low total abundance, but this is likely because NOAA's CMAP cruise was cut short due to the hurricanes, but just has fewer dominant families compared to summer. Winter was dominated by cyanids and triglyphids, mostly Atlantic and spot croaker and various sea robins accounting for that. This suggests that winter spawning is taxonomically concentrated between these families. So spring appears to be more of a transitional season with a good amount of eggs, and it has a mixed bag of contributors. Our cap ordination shows strong seasonal differences in fish egg assemblage composition. The first canonical axis primarily separates summer from winter samples, while the second axis distinguishes fall from spring. So summer assemblages, which are your blue-green crosses, are tightly clustered, indicating consistency across samples, whereas winter, which are your purple X's and green triangles, show greater variability. Now, I want to point out some more zoomed-in species level findings. 137 of our eggs came from these 6 species, and these species are a part of the Gulf Reef Fish Management Plan that's managed by NOAA. The highest abundances were from vermilion snapper and red snapper, but I do want to talk about hogfish in particular for just a second. Going back to the table that I showed earlier, we can see that hogfish is known to spawn in winter, spring, and early summer months. So this species was never recorded in the August to September CMAP samples. I detected it for the first time in our June 2024 sampling period, which is when the spawning period for hogfish begins to taper off. 3 eggs were collected from 3 sites on the West Florida Shelf in summer 2024, and 1 egg was collected in spring of 2025. And this is important because we're adding new species to our inventory, but this is just one pretty big example. So this analysis of our first year of sampling will play into our first targeted study of Shelf Phase 3, which is looking at the seasonal dynamics and spawning on the West Florida Shelf. Fish egg beta diversity differed significantly among seasons based on the data that I analyzed, with season explaining about 8% of the community variation as shown by Permanova testing. Finally, some ideas for the future. There appears to be some seasonal influence on spawning patterns in my data from the first year. But will we see it again in the second year data? I'll have broader spatial coverage in fall 2025 than I did in fall 2024. So it'd be really interesting to get the full picture, like on a stack bar chart, like I showed earlier. And because our sampling stations are fixed and geographically close, communities at nearby stations may be more similar just simply due to spatial proximity. So I'd like to evaluate whether spatial autocorrelation was present in the data. I suspect that it is. I'd also like to test an effect of region and depth on our communities as well. So for future outlooks, the Shelf project trends forward with continuing the time series, which will make up the first chapter of my PhD. We've completed all but 1 season of collections. The last Shelf cruise is this March. Let me know if you're interested in joining or collaborating with us. We've had a lot of collaborations with some labs here. We've had some ChemOsh people, some PhysOsh people join us, collecting their data simultaneously with us. So we're always open to collaborating. I wanted to thank all the organizations that have supported my projects and me personally, and thank you all for coming to my talk today. And now I'll take questions and comments.
Everyone, thank you so much for coming to my talk today. Emma, thank you for the introduction. As she said, my name is Layne Leggett. I'm currently working with Dr. Smith-Morowski, and I'm going to be talking today about the work I've been doing so far for my master's thesis, also known as the Barnacle Baseline Project. And specifically today, I'm going to be talking about the methods that I've developed so far. So our goal with this project is to assess contaminants of emerging and known concern, which you heard a little bit with AJ's wastewater talk earlier, but we want to look at those within Tampa Bay using Acorn Barnacles as our bioindicator. So before I can kind of dig into the Barnacle Baseline Project, I feel like I needed to tell you all about, if you haven't heard already, a much larger and complex project that the Barnacle Baseline is a part of, and that is the Tampa Bay Surveillance Project. So the goal of TBS is to study CECs and legacy pollutants in sediments, benthic invertebrates, fish, and wastewaters within Tampa Bay, as you heard earlier with AJ. There's also a whole human aspect of the project where there's subsistence fishing surveys to better understand the communities that could be affected by the regular consumption of the project's fish species. So what fish are we specifically looking at? There is the snook, the red drum, the spotted sea trout, and the sheep's head. And then we are looking at barnacles and oysters as our invertebrates of interest. So just a little bit of background. Again, if you were here for AJ's talk, he did talk about CECs a little bit, but if you don't know, contaminants of emerging concern, CECs, according to the EPA, are pollutants for which there's limited data on their occurrence, environmental fate, and toxicity. And those can include some PFAS compounds, pharmaceuticals, and phthalates, for example. And then you also have CECs, or contaminants of known concern, like banned pesticides like DET and other toxic metals, for example. So how do barnacles fit into TBS's goals? So they're sessile adult stage, meaning that they stay in the same spot once they settle in their adult life. They're there forever. So this could give us potential to find possible sourcing of the pollutants and where they might be coming from. And also predation from animals such as sheep's head can help us get a better idea of the biomagnification of these contaminants. So just some overall background on the Barnacle Baseline Project. It is funded by South Carolina Sea Grant as a smaller companion project to the Tampa Bay Surveillance Project. And we are focusing on regional and seasonal differences in CEC and CKC uptake. So we are specifically going to be looking at barnacles in the wet season and the dry season and seeing if their chemicals and their chemical contaminants are different. And we are looking at 17 sites around Tampa Bay. And again, their sessile adult stage and fast growth rate make them a good fit for these site-based studies. So we do have some main questions that we're looking to answer. And first, it would be, where are the CECs and CKCs in Tampa Bay coming from? So what kinds of CECs are found near wastewater treatment outfalls and storm drain outfalls? And do these two outfall types possibly carry different contaminants? Where do these contaminants accumulate most in the bay? So are the concentrations in these barnacles varying by location, by outfall type, and do these concentrations vary over time? And also, how does rainfall, mostly probably influenced by climate change, how does that influence contaminant levels in the bay? So do barnacles from Florida's wet season have an increased contaminant level than barnacles from the dry season? So as you can see here, if you don't know Florida's wet and dry season, from October to around the end of May is our dry season, and we enter our rainy season from June to September. So how are we looking to answer these questions? First, we had to manufacture what we are calling our barnacle arrays so that we could actually collect and sample these barnacles. And that involved cutting a lot of PVC pipe to actually build the structure itself. Drilled 4 holes in each glass tile. If you've never drilled glass before, it's a very fun job to do. So glass was chosen as an inert recruitment substrate because it could be easily sampled and in theory would not have a lot of these contaminants that we're looking for. And then I also had to drill holes into the PVC arrays for zip tie attachment. And then we secured each tile into the array using the zip ties, zip tying them on there so that we could easily take them on and off board collection. And that ended us with a 3x3 grid of glass tiles. And each side, it is 2-sided, so it's a 3x3 on both sides. So how did we know that that design was going to work? Well, we did have a test site out in Bayboro Harbor. It's actually still out there to this day because it is now going to be one of our sites. But this is how we test the structure and kind of seeing if it was going to work. And I do take weekly photos and collect the temperature data from this array every week. So it helps us to show the growth patterns for both the wet and the dry season. So if you look here, this one here, that is the 2nd week that the array was ever out at the start of the wet season. And this was the week right
this was actually the day that I collected the wet season tiles. So this is how much growth had occurred over the wet season. And then this was the week following when I replaced those tiles for the start of the dry season. And this was just last week. So this is what our dry season tiles are looking like in Bay Royal Harbor. It also is helping to kind of show predation and overgrowth of other species that could outcompete them for space. So a lot of these, it's not easy to see, but a lot of them are, especially the ones that are staying on there, are getting very overgrown with oysters, and the oysters are starting to even outcompete the barnacles. So it's showing that as well. And then it's also showing a competition between ivory and striped barnacles, which is the two primary species that we're seeing. So these are our deployment locations. Now that I built all the 17 structures, we were able to actually place them out in the bay. Here you can see our 17 deployment locations. This one here at Egmont Key actually no longer exists. It was originally deployed, but because of the wave action out there, that site was lost. So we are left with 16 sites right now. And several of these sites are co-located near TBS oyster sites. So we are looking at oysters in these similar areas as well. And then those are also areas where the TBS fish were caught. And several are near wastewater treatment outfalls. And then if you look at this map here, these are some of the stormwater discharges known around Tampa Bay. So now that I built all my structures, we can go into the installation and collections. We installed at all of those sites that I just mentioned, but each of the arrays were installed with a do not disturb sign so that if someone did come across it, especially fishing, or if they were just in the area and they wanted to know what it was, they do have a do not disturb sign on them with the QR code to the TBS and Barnacle Baseline website to learn more about the work. They all have paddle tag labels to identify what columns are which, and then they also have air tags and hobo temperature sensors. So every 5 minutes, the water temperature is being taken. So 2 of the 3 columns were collected and replaced at the start of the dry season in October. So once we went out in October, I essentially chose 2 columns to take, and then 1 is going to be staying for the entire year so that we can see if that full year is going to have possibly different contaminant loads than just the wet season or the dry season barnacles. And these are 2 of the arrays and what they looked like at the conclusion of the wet season. So I mentioned that they all have air tags on them. So this is kind of a goal of ours to be able to monitor them over the full year of sampling. Essentially, the air tag is a little interesting. It has to be fully out of the water, and an iPhone or any Apple product has to be in the area to actually give it a signal to send to my phone that this is the last time that that air tag has been seen. So it does help with monitoring a little bit in terms of I can look at my phone right now if I wanted to and see the last time that each of these were seen. It does get a little weird at times because McKay Bay, not a lot of people are fishing up there or really in that water. So that one doesn't get a lot of pings. So it hasn't been seen for a while, but we hope that it's still there. And then this one, it usually gets a lot of pings, but we had one of our fishing captains actually tell us that they don't know if it's still there or not. So we actually are doing some oyster sampling next week. So that is the other ways that I can kind of get out there and actually put an eye on them to make sure that they're still there is when we go out, because the oyster sampling is a little bit more frequent than the barnacle sampling. So I'm able to at least a few times put eyes on them, but these air tags really help with monitoring them over the full year. So after the collections, they're brought back to the lab and I'm photographing all of them for % cover and count analyses. And it's really hard to get algae and other things off of them. So without introducing more contaminants to the tiles. So I actually am doing a rough count of all the barnacles on each tile, and then also differentiating between the 2 species that I mentioned. We have the striped barnacles and we also have the ivory barnacles. So I am taking rough counts of those as well. And then I weigh the total biomass. So a lot of times we're finding little crabs and algae and oysters as well on these tiles. So I am weighing the total biomass and then I am weighing just the barnacle biomass from each tile. And then they are dissected out of the shell. If you didn't know what the inside of a barnacle looks like, it's right there.
They are crustaceans, so essentially it's a lot easier to dissect a barnacle than you may think. You just open the operculums and pluck out the body. It's pretty simple. And then the smaller ones, their base plate hasn't really formed yet, so you can flip them over and pull the body out that way. And then barnacle tissue, there's not a lot of it and they're very small. And for some of the laboratory analyses, we are going to have to freeze dry the tissue. So in order to have enough tissue, all of the barnacle tissue from all of the tiles from that each site are composited into one jar for the later contaminant analyses. So what are we noticing so far? The striped species dominate over the ivory species in the wet season. And overall growth is extremely variable by site. So some sites who were placed later in the wet season had more biomass than sites that were placed at the very beginning of the wet season. And site tissue mass so far, the totals are ranging anywhere from about 0.8 grams all the way up until 25 grams of tissue. So next step, I still have to complete the last few dissections for the wet season and begin the contaminant analysis for those samples, and then process those tile images that I was taking for the wet season. And then in May, we're going to be collecting the dry season tiles as well as those tiles that have been out there for a full year. And so I'm then going to be completing these same dissections and methods for the dry and year tiles to ultimately test for CEC concentrations by site and by time within these barnacle tissues around Tampa Bay. And with that, I will take any questions you might have.
Good afternoon, everyone. Last talk. Let's make this quick because there is a TGIF after this. So I'm going to be presenting on a novel sensor that I've been working in development in a conjunction with COT as base of the NOAA project and a local company based out of Clearwater called Connect 6 that does cloud salometers. So how they're moving from cloud salometers to marine is an interesting story. Okay, so the purpose of the research, if anyone's used scientific instrumentation, you know that sometimes it is prohibitively costly. So we're trying to set out first to make a time of flight lidar that can measure fluorescence, but at a much cheaper cost. The second that we can use is the lidar provides a distance component that not a lot of fluorometers right now really have. So you can kind of look out into the water column. What this can provide us is crucial information about the phytoplankton biomass, which then can allow you to start thinking of biogeochemical cycles, primary production, and a HAB dynamic, mainly with red tide, because we all live in Florida, so it's a very important thing to us. So I'm just going to give a little primer. I know not everyone has optics background, totally fine. So how does fluorescence work? Any molecule like chlorophyll, a chemical like argon or neon absorbs light at one wavelength and then will emit at a different wavelength. The one that we're really primarily concerned is chlorophyll A. It mainly absorbs in the blue spectrum of the high energy and then will emit red light. Fluorescence can provide a lot of information, such as health of the algal cell, Fe over Fm. I'll be more than happy to give more talks about that, but it's kind of how happy they are. It's more of a quantitative measurement, not really a qualitative, but it can also be used to determine biomass. A lot of remote sensing will do chlorophyll fluorescence and then kind of backtrack into biomass. So how do time of flight sensors operate? Essentially, they pulse out flashes of infrared light, and then they measure how long that light takes to return. If anyone has a newer car that has parking assist and whatnot, it's the same technology. When the car yells at you that, hey, you're too close to something, it's that similar technology. What's really nice about this is that it can improve the signal to noise ratio. What that means is anything that's electronic generates noise. So any instrument you have, when you're measuring something, there's some noise in there because of just electronic read. This technology allows you to essentially cut that noise out and you're getting a stronger signal, which is really important when you're trying to find stuff like fluorescence and such. And then it can also collect photons of light over many pulses, so you can actually time your pulses to get a better signal. And then it can provide that depth and distance. So I can pulse out a lot of photons and then get a lot back, and then you can kind of work out the distance component to it. So here's the sensor concept. So essentially, it's going to have So it's going to have a pulse light of blue, and then obviously in its field of view, it has a detector that's going to detect that that red fluorescence or that red emission, like I was saying. Obviously, in the water column, you're going to have ambient light, so that's going to kind of mess with your measurements a bit, just because not only does algal cells fluoresce, but also color dissolved organic material in the water column. We like to refer to CDOM, or as it's now being called FDOM, that can also fluoresce. But the nice thing is that when you have this distance component, because of the LiDAR, you can look here, but you can also look here. That's really important because if you were to put this, say, on a mooring and such, if you're staring out into the water column, you can potentially catch a harmful algal bloom as it's potentially initiating, or you can use it as another way to kind of tease out fluorescence and such. So here are our prototypes. I'm going to be talking about two. This is the single pixel. What a single pixel just means is that it's looking at a single pixel in its view field. This is a multi-pixel that can provide a better resolution image. This is what we're using as a fluorometer with also a little bit of distance, but this can really give the better distance. Little small disclaimer here. Both sensors, but the Hydra can provide the better resolution, and this one's a lot cheaper. So this is like $10 to $50. This one's still a little bit more pricey at about $1,000, but still significantly less than any fluorometer that you currently find on the market. So the methods that we used, so we're adapting our calibrations from the YSI. YSI makes these really cool sondes. The P-Bond project has them all out on their moorings. So they use what's referred to as rhodamine to do their dilution calibrations. Rhodamine is very stable, but it can fluoresce, very akin to chlorophyll A. So we kind of use rhodamine in our sensor development. We also use, so when you're developing a new sensor, you're just trying to figure out what can it do and what is the signal that we're getting. So we also kind of took some bay water out of Bay Burrow and used that as a dilution series, which I feel really bad for the phytoplankton in there because they're being dumped into fresh water. At the end of the proof of concepts, we used exopyridinium from FWC to test the single pixel's capability on live algal cells, as well as the HAB species. Then we also did additional testing to find the internal temperature. We were noticing that the internal temperature in this device was causing a drift in the measurement, so we wanted to try to eliminate that as part of the development process. And then we've also done some seawall deployments out here off of Bay Burrow, and we've used fluorescent paint trying to characterize saturation. What saturation just means is that if you take a sensor and you shine it at something and it just completely whites out, that's saturation. These fluorometers can also saturate if there's too much. So we wanted to try to know what was the saturation limits. And then we now have 4 functional units that can be run in sequence to try to characterize. So, try to get out all instruments will technically have some quirk to them, but trying to characterize them in part of our development process has been a fun task. So I'm going to first tell you, this is not very pretty data. This is data based off of some research and development that we've been doing. But this one, if I can draw your attention to, is we are using a wet labs and our sensor. And this is based off of a 1 second average to ours. Now, you would say that, okay, ours is a bit more noisier, which you are correct. You see those peaks, but as the dilution starts coming in, you start to get a better resolution. And then we start seeing that the wet labs will overestimate where we'll get a better estimation, because the wet lab sensor can saturate at a lower concentration than ours. That helps just because, once again, part of the goal of this project is trying to build a cheaper fluorometer that can be used. So this were the 2s that we did. This was part of the phase 1 testing. And then here's the pyridinium test. So we kind of did our, it gets a bit noisy just because it's pulsing multiple pulses of red light. So it's going to be a little bit noisier at lower concentrations. Obviously, you can see that the wet labs is having a similar problem, but as we start to get a higher concentration of pyro, there's a nice, almost a nice agreement in some respects with ours now showing a bit better sensitivity at the higher end than the wet lab. So we were really happy. And then the really cool thing about what the Hydra sensor does is how I said it gives a better resolution. This is rhodamine, but we had that like at one end of the bench top. So we had the sensor here, and where like Patrick is sitting is where we had the rhodamine, and it could see, you can see that nice fluorescent signal. So you can get that distance component to it. So we were really happy about it. This for the pyrocells, the final edition was about 270,000 cells per liter. So we tortured some pyridinium for here, but it was worth it because it gave us really interesting results. So future work that we're trying to do is, obviously we have seen that there's a little bit of noise due to ambient fluorescence. So currently I'm working on a Python processing pipeline to try to apply a correction to try to tighten the data up a little bit. Because we have 4 additional units, we're planning on future field tests with NOAA off of the Beers Calval cruise that's coming up in May. FWC is very interested, so is Moat Marine and Tampa Bay Watch, just because, once again, cheaper fluorometer can provide that. We're also going to be developing an IOP package. What IOP is just water's inherent optical properties, so we're just trying to make, a better in-water optics and such. And then ultimately, we can, down the road, we want to then compare this fluorometer's measurements to that of maybe a remote sensing for like the Veers satellites, Remotus, Aqua, and Terra for validation, which is something that NOAA is very interested in on this cruise because they do Veers validation. So if we have a cheaper fluorometer that we can now try to have it as a validation, that'll be really cool. And I'm curious to see that data. So I just want to give a special thanks to my collaborators. A lot of them, if you ever sit on one of our meetings, we're going in every which direction with this sensor, but we've come up with a lot of really good ideas. I think the most recent idea is trying to use this technology for an optical CTD, which I have some quibbles about, but we're trying to essentially see what avenues we can use this technology for. And I'm also very, a special thanks to Florida High Tech Corridor, which has more or less carried the rest of me through my degree. So I'm very happy about that. So references, thank you. And any questions?